Hello students, welcome to Karnataka. This is Basaraj sir, your biology master teacher. So students, today's class is about your monohybrid cross and dihybrid cross. ಒಂದು ಚಾಪ್ಟರ್ ಇದೆ ನಿಮ್ಮ ಟಾಪಿಕ್ ಅಲ್ಲಿ, ನಿಮ್ಮ ಸಿಲೆಬಸ್ ಅಲ್ಲಿ, ಪ್ರಿನ್ಸಿಪಲ್ಸ್ ಆಫ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಅಂಡ್ ವೇರಿಯೇಷನ್. ಈ ಚಾಪ್ಟರ್ ಇಂದ ಎವ್ರಿ ಸಿಂಗಲ್ ಇಯರ್ ನಿಮ್ದು KCET ಎಕ್ಸಾಮಿನೇಷನ್ ಅಲ್ಲಿ ಅಟ್ ಲೀಸ್ಟ್ 3 or 4 ಕ್ವಶನ್ಸ್ ಬಂದೇ ಬರುತ್ತೆ. ಅಟ್ಲೀಸ್ಟ್ 3 ಕ್ವಶನ್ಸ್ ಬಂದೇ ಬರುತ್ತೆ. ಅದರಲ್ಲಿ ಒಂದು ಸ್ಮಾಲ್ ಟಾಪಿಕ್ ಇದೆ ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ monohybrid cross and dihybrid cross. ತುಂಬಾ ಸಿಂಪಲ್ ಟಾಪಿಕ್, ತುಂಬಾ ಸಿಂಪಲ್, ಬಟ್ ಅಲ್ಲಿನ ಕ್ವೆಶನ್ಸ್ ಬರೋದಲ್ಲ, ಆ ಕ್ವೆಶನ್ಸ್ ತುಂಬಾ ಟ್ರಿಕಿ ಆಗಿರುತ್ತೆ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ನಿಮ್ಗೆ ತೋರಿಸ್ತೀನಿ ನೋಡಿ. ನಿಮ್ಮೆಲ್ಲರಿಗೂ ಗೊತ್ತು monohybrid cross dihybrid cross ನೀವು ಮಾಡಿದೀರಾ ನಿಮ್ಮ ಸಿಲೆಬಸ್ ಎಲ್ಲ ಕಡೆ. ಬಟ್ ಕ್ವೆಶನ್ಸ್ ನೋಡಿ ಹೆಂಗ್ ಬರುತ್ತೆ. ಕ್ವೆಶನ್ಸ್ ನೋಡಿ, ಈ ತರ ಬರುತ್ತೆ ಕ್ವೆಶನ್ಸು, ಈ ತರ ಬರುತ್ತೆ ಕ್ವೆಶನ್ಸು, ಇಲ್ಲ ಅಂದ್ರೆ ಸಿಂಪಲ್ ಆಗಿ ಈ ತರ ಬರುತ್ತೆ ಕ್ವೆಶನ್ಸು. ನೋಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್, ಸೊ ನಿಮ್ಗೆ ಫಸ್ಟ್ ಈ ಕಾನ್ಸೆಪ್ಟ್ ಅರ್ಥ ಮಾಡಿಸ್ತೀನಿ ಇವತ್ತು. ಅದಾದ್ಮೇಲೆ ನಾವು ನಿಮಗೆ ಕ್ವಶನ್ ಪ್ರಾಕ್ಟೀಸ್ ಕೂಡ ಇವತ್ತಿನ ಕ್ಲಾಸಲ್ಲೇ ನಿಮಗೆ ಮಾಡಿಸ್ತೀನಿ. ಓಕೆನಾ? ಸೊ ಎಲ್ಲರೂ ಸ್ಟೂಡೆಂಟ್ಸ್ ಕ್ವಿಕ್ಲಿ ವಿಡಿಯೋನ ಲೈಕ್ ಮಾಡಿ ಒಂದ್ಸತಿ, ಆಮೇಲೆ ವಿಡಿಯೋನ ಶೇರ್ ಮಾಡಿ ಎಲ್ಲರಿಗೂ ಹೂ ವಾಂಟ್ಸ್ ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ವಾಟ್ ಎಕ್ಸಾಕ್ಟ್ಲಿ ಇಸ್ ಯುವರ್ monohybrid cross and dihybrid cross. ಆಮೇಲೆ ಈ ಕಾನ್ಸೆಪ್ಟ್ ಇದೆಯಲ್ಲ, ಈ ಕಾನ್ಸೆಪ್ಟ್ ನಿಮ್ದು ಬೋರ್ಡ್ ಎಕ್ಸಾಮಿನೇಷನ್ ಕೂಡ ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟು. ಇಫ್ ಯು ಗೋ ಚೆಕ್ ಔಟ್ ಎನಿ ಪ್ರೀವಿಯಸ್ ಇಯರ್ ಪೇಪರ್, ಯಾವುದೇ ಪೇಪರ್ ತೆಗೀರಿ ಪ್ರೀವಿಯಸ್ ಇಯರ್, ಅಟ್ ಲೀಸ್ಟ್ ಒನ್ ಕ್ವೆಶನ್ ನಿಮ್ದು monohybrid ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಕ್ ಹೇಳ್ತಾರೆ, ಆರ್ ಎಕ್ಸ್ಪ್ಲೇನ್ dihybrid cross ನ ಸಿಂಪಲ್ ಕ್ವೆಶನ್ ಇರುತ್ತೆ. ಆರ್ the question will be asked as what is law of dominance, what is law of segregation, and what is law of independent assortment. ಸೊ ಈ ಎಲ್ಲ ಕಾನ್ಸೆಪ್ಟ್ಸ್ ನ ನಿಮ್ದು KCET ಆಗಿರ್ಬೋದು ಮತ್ತೆ ಬೋರ್ಡ್ ಎಕ್ಸಾಮ್ ಆಗಿರ್ಬೋದು, ಎರಡು ಕೂಡ ಈ ಚಾಪ್ಟರ್ ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟು, ಈ ಸೆಷನ್ ಕೂಡ ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟು. ಸೊ ವಿತ್ ದಟ್ ಬೀಯಿಂಗ್ ಸೆಡ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ಲೆಟ್ಸ್ ಕ್ವಿಕ್ಲಿ ಸ್ಟಾರ್ಟ್ ದ ಕ್ಲಾಸ್ ವಿತ್ ದ ಬೇಸಿಕ್ಸ್. ಈ ಚಾಪ್ಟರ್ ಬೇಸಿಕ್ಸ್ ಏನು, ರೈಟ್? ಈ ಚಾಪ್ಟರ್ ಅಲ್ಲಿ ನೀವು there is a one main scientist who is also called as the father of genetics. ಯಾರಾದ್ರೂ ಹೇಳ್ತೀರಾ ಯಾರಂತ? ಯಾರಾದ್ರೂ ಹೇಳ್ತೀರಾ ಚಾಟ್ ಅಲ್ಲಿ? Who is known as the father of genetics? Anyone? The father of genetics is none other than the Gregor Mendel. He is none other than the Gregor Mendel, students. Gregor Mendel was an amazing biologist. ಆಮೇಲೆ but starting ಅಲ್ಲಿ he was a mathematician, then he became a biologist. He is now Gregor Mendel. Okay, Gregor Mendel. Now students, ನೀವು ಅರ್ಥ ಮಾಡ್ಕೋಬಹುದು, ರೈಟ್? ನೀವು ಗ್ರೆಗರ್ ಮೆಂಡೆಲ್ ಅಲ್ಲಿ ಒಂದು ಫೇಸ್ ಫಸ್ಟ್ ಕ್ವಶನ್ ಬರುತ್ತೆ, ವಾಟ್ ಡಿಡ್ ಹಿ ವರ್ಕ್ ಆನ್? ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ನಿಮ್ದು ಡ್ರೋಸೋಫಿಲಾ ಮೆಲನಿಗೆಸ್ಟರಾ, ಪೀ ಪ್ಲಾಂಟ್, ಯಾವ ಪ್ಲಾಂಟ್ ಮೇಲೆ ವರ್ಕ್ ಮಾಡಿದ್ದಾನೆ. ಓಕೆ, ಸೊ ದರ್ ಇಸ್ ಅ ಪ್ಲಾಂಟ್ ಕಾಲ್ಡ್ ಎಸ್ ಪೈಸಮ್ ಸಟೈವಮ್. ನಿಮಗೆ ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ ಅವರು ಸಮ್ ಟೈಮ್ಸ್ ಏನ್ ಮಾಡ್ತಾರೆ ಅಂತಂದ್ರೆ ಪೀ ಪ್ಲಾಂಟ್ ಕೊಡಲ್ಲ, ಸೈಂಟಿಫಿಕ್ ನೇಮ್ ಹೇಳ್ಬಿಡ್ತಾರೆ, ಸರಿನಾ? ಪೈಸಮ್ ಸಟೈವಮ್. ಪೈಸಮ್ ಸಟೈವಮ್ ಅಂದ್ರೆ ಪೀ ಪ್ಲಾಂಟು, ಅಂದ್ರೆ ಪೀ ಪ್ಲಾಂಟ್. ಇದು ಏನಿದು? ಪೀ ಪ್ಲಾಂಟ್. ಸೊ ಈ ಪೀ ಪ್ಲಾಂಟ್ ಅಂತ ಒಂದು ಪ್ಲಾಂಟ್ ಮೇಲೆ ಮೆಂಡೆಲ್ ಕೆಲಸ ಮಾಡಿದ್ದ. ಓಕೆ, ನಾವು ಅದರ್ ಕ್ವೆಶನ್ ಇಸ್ ಫಾರ್ ಹೌ ಮೆನಿ ಡೇಸ್, ಫಾರ್ ಹೌ ಮೆನಿ ಮಂತ್ಸ್? ಗೆಸ್ ಮಾಡ್ತೀರಾ ಯಾರಾದ್ರು? ಹೌ ಲಾಂಗ್ ಡು ಯು ಥಿಂಕ್ ಮೆಂಡೆಲ್ ವರ್ಕ್ಡ್ ಆನ್ ದಿಸ್ ಪ್ಲಾಂಟ್? See, all of you preparing for KCET, you will prepare for 1 month, 1 year maximum. Or if you want to take a break here, again 2 years, let's say 2 years. But Gregor Mendel was very determined. He worked for almost 7 long years. Gregor Mendel worked for 1856 in the 1863. That is 7 long years. 7 years he worked on this experiment, that is hybridization experiment. And students, in this experiment, what did he take? In his experiment, he took something called as 7 pair of characters. 7 characters ಅಂತ ತಗೊಂಡಿದ್ದ ಅವನು. ಈ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಅಂದ್ರೆ ಏನಪ್ಪ? ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಅಂದ್ರೆ ಏನು? ಹೈಟ್ ಆಗಿರ್ಬೋದು, ಕಲರ್ ಆಗಿರ್ಬೋದು, ಫ್ಲವರ್ದು ಕಲರ್, ಫ್ಲವರ್ ಪೊಸಿಷನ್, ಓಕೆ, ಪ್ಲಾಂಟ್ ಹೈಟ್, pod color, seed shape, all of these are your characters. ಅದೇ ಸೀಡು ರೆಡ್ ಸೀಡ್, ಗ್ರೀನ್ ಇದೆಯಾ, ಎಲ್ಲೋ ಇದೆಯಾ, that becomes a trait. ಅರ್ಥ ಆಗ್ತಿದೆಯಾ ಎಲ್ಲರಿಗೂ? ಸೊ there is a difference between character and trait. ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. All of these are called as your characters. Stem height, flower color, flower position, pod shape, pod color, seed shape, seed color, all of these are called as your character. Then what is a trait? Trait is nothing but the alternative variation. ಇದು ನೋಡಿ, ಇಲ್ಲಿ tall dwarf, violet white, all of this becomes trait. This is a character. So Mendel worked on 7 different characters on the pea plant. 7 characters, 7 long years. Imagine 7 characters, 7 long years. Mendel worked. Mendel worked for 7 long years. Students, he took something called as pure lines. Pure lines tagondida, where he did the work. Now students, ನೀವು ಅರ್ಥ ಮಾಡ್ಕೋಬೇಕು. What do you call this monohybrid and dihybrid? What is the actual name of this experiment? The name of this experiment is called as hybridization. It is called as what? Hybridization. ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ ಎಲ್ಲರೂ. ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್ ಇದು. It's called as hybridization experiment. Hybridization experiment. Now, what exactly are the steps in hybridization experiments? There are 3 main steps. How many are there? There are 3 main steps in hybridization experiment, which was done by the Mendel. It was done by the Mendel. Now, first step is called as selection of parents. It is called as what? Selection of parents. First step ಅಲ್ಲಿ ನಾವು parents select ಮಾಡ್ತೀವಿ. Okay, selection of parents. ಈ parents ಯಾವತ್ತು true breeding lines ಮಾತ್ರ ಇರಬೇಕು. True breeding lines. Okay. Now, second step is called as your crossing. It's called as cross pollination. Cross pollination. Cross pollination. ಇಲ್ಲಿ ನಿಮಗೆ F1 ಅಂತ ಸಿಗುತ್ತೆ, ಒಂದು F1 ಸಿಗುತ್ತೆ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಮಾಡಿದಾಗ. The third step is called as selfing of F1. It's called as selfing of F1. Okay. So 3 main steps in hybridization. ಈ 3 main steps ನಾವು monohybrid cross ಅಲ್ಲಿ, dihybrid cross ಅಲ್ಲಿ ಕೂಡ ಯೂಸ್ ಮಾಡ್ತಾ ಇದೀವಿ. ಓಕೆನಾ? So let's get into the first cross, that is called as your monohybrid cross. ಅದಕ್ಕಿಂತ ಮುಂಚೆ this thing is very important, as you know. There's 7 different characters. ಇದರಲ್ಲಿ ಕ್ವೆಶ್ಚನ್ ಹೆಂಗ್ ಬರುತ್ತೆ ಅಂತಂದ್ರೆ, they can ask you which of the following is dominant. ಕೇಳಬಹುದು, which of the following characters is dominant, which is recessive. ಅದಕ್ಕೆ ಒಂದು ಸ್ಮಾಲ್ ಟ್ರಿಕ್ ಇದೆ. The trick here is gravity. The trick here is called as gravity. Gravity ಅಂತಂದ್ರೆ each individual letter here is representing a dominant trait. Dominant trait. For example, here the green represents the G. The G is for green, that is pod color. It is for pod color. Pod color. Green. Now what is R again? R is for seed shape. Round seed shape. Seed shape round is dominant. Now what is A? A is your flower position. A is your floral position, that is axial. A for axial. 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 Then what is V? V is your violet, that is flower color. Can you see? Flower color, violet. Flower color. Flower color, that is violet. Then what is I? I is basically inflated, that is pod shape, inflated. I is pod shape. Pod shape. Now what is T? T is your stem height, tallness. Tallness ala stem height. Now what is Y? The Y stands for yellow. That is seed color. Is seed color. Seed color. So ಇದು ಎಲ್ಲ ಡಾಮಿನೆಂಟ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್. ಇದು ಎಲ್ಲದರದ್ದು ಕೂಡ ಡಾಮಿನೆಂಟ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ನ ಗ್ರಾವಿಟಿ ಅಂತ ಪದದಲ್ಲಿ ಯೂಸ್ ಮಾಡಿದೀವಿ. So gravity, that is each individual one is called as your dominant trait. That is green, round, axial, violet, inflated, tall, yellow. Clear? Clear, students? Okay. This is called as your characters, dominant traits. Dominant traits. In examination, they can ask you any one of the following. They can ask you. Okay. So with that being said, let's start with something called as your monohybrid cross now. Start something called as monohybrid cross. Monohybrid cross. And students, remember, in examination, in your board examination, you should follow the same method which I am teaching you. Follow the same method in the examination also. It will be very, very useful. Same method you follow. The first thing which you need to write down here is the parents. The first step is writing the parents. Parents. First step is writing the parents. Okay. First parent is going to be homozygous. Homozygous tall. Homozygous tall is capital T, capital T. Homozygous is the same, tall. The other parent is going to be homozygous recessive. Homozygous recessive. That is small t, small t. These are the parents. For example, this is the dominant. This is the dominant one. This is the recessive allele. Dominant alleles, recessive alleles. Okay, dominant alleles and recessive alleles. The next step is forming the gametes. Next step is forming the gametes. So students, in examination, if you want to marks, every single step is important. Now here we will get a single gamete, capital T. Here we will get a single gamete, that is small t. Next step is called as crossing. 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 That is crossing here. Okay. When you do the crossing of your gametes, we get a F1. F1. A F1 is going to be capital T, small t. Capital T, small t. And this capital T, small t is heterozygous. But since capital T is dominant here, the phenotype, the phenotype is going to be tall here. The phenotype is going to be completely tall here. The phenotype is tall here. Okay. That is called as law of dominance. So students, from the monohybrid cross, we are getting two main laws. The first law is called as law of dominance, and the second law is called as law of segregation. This law kuda namge monohybrid cross na sigutte. Clear? Then what about dihybrid cross? From dihybrid cross, we will get something called as law of independent assortment from dihybrid. Okay. Right now, this is monohybrid cross. So we get law of dominance from here. Law of segregation also is here. I will explain that later on. Okay. Now, next thing is called as selfing of F1. Next step is called as what? Selfing of F1. Selfing of F1. So this is F1. Capital T, small t. Capital T, small t. Okay. Now, what again we find gametes. Again we find gametes now. Gametes are going to be here. Capital T, small t. Gametes, capital T and small t. Okay. Gametes are here. Okay, gametes are here. Now students, next step is crossing over. Next step is crossing over. In next step crossing over madabekadre, there is something called as Punnett square we use. Something called as Punnett square. Understand what is Punnett square? Draw and show you. See, we will draw Punnett square here. Okay. What do we have gametes here? Capital T, small t, capital T and small t. Okay. Gametes are set. Now, capital T, okay. Capital T, small t, capital T, small t, small t, small t. This right here is called as a Punnett square. It is called as your Punnett square. Many questions will come from here also. Simple question can come. Punnett square. Now students, from this Punnett square, now we need to write something called as phenotypic ratio. Phenotypic ratio and genotypic ratio. Phenotypic ratio as well as genotypic ratio. Okay, now here this is going to remain as tall. Why? Both are dominant alleles. It's going to remain as tall. Heterozygous condition. Capital T is dominant. So capital T is for tallness here. This is tall. This is a dwarf. So here also we'll get what? Here we will get tall again. Here also we will get tall. We will get tall here. Now this one, both are your small t. This is just like your parent. This is going to be dwarf, means short. It's going to be dwarf. Now tell me students, if you look at the phenotypic ratio means how a plant looks from the outside. Aache in the plant yang kaanasta ide. That is called as your phenotypic ratio. Aache in the kaanasta ide. Tall. 3 is 2. One dwarf, single dwarf aache in the nodavikke. Then what is genotypic ratio? Genotypic ratio basically means what is the category which we find here. That is it homozygous? ಎರಡು same ಇದೆಯಾ? Heterozygous. This is homozygous dominant. This is homozygous recessive. Homozygous means ಎರಡು ಒಂದೇ ತರ ಇದೆ ನೋಡೋದಕ್ಕೆ ಎರಡಲ್ಲಿನೂ ಕೂಡ. Heterozygous means both alleles look different from each other. Okay. Now this is homozygous dominant, a single one. These two are heterozygous dominant, 2. Homozygous recessive, 1. So the phenotypic ratio is going to be 3:1. At the same time, genotypic ratio is going to be 1:2:1. Students, in board examination, till now, if you write, you will get out of 5 marks question, 4 marks easily. 4 marks easily. 1 more mark will be for explaining the law of segregation or law of dominance, any one of the question. So students, I believe till now there should be no doubt in the monohybrid cross on the board right now. And students, if you want these notes will be available on the Telegram channel. All of you are aware, right? In Telegram channel, all the notes will be updated there. Okay. Now students, this is your monohybrid cross. Now as I told you, this is your Punnett. This is what is given in your book. Tall, dwarf. 3:1 ratio of phenotypic, 1:2:1 genotypic ratio. Okay. Now students, in examination they can ask you for 1 mark question also. What is law of dominance? Or they can ask you what is law of segregation. So I will tell you right now what exactly you need to write for that also. Okay. For law of dominance, there are 3 main points. What are 3 main points? Characters are controlled by discrete unit called as factors. Factors is basically your alleles here, okay, or gene in many cases is very interchangeable. Okay, so all the characters, tallness agirbodu, dwarfness agirbodu, everything is controlled by your alleles, controlled by gametes, okay, or genes. Factors occur in pair, dominant recessive, capital T or capital T small t. Now in a dissimilar pair of factors, what do you mean by dissimilar? ಎರಡು ನೋಡಕ್ಕೆ ಒಂದೇ ತರ ಇಲ್ಲ. That is capital T small t. In a dissimilar pair of factors, one member of the pair dominates over the other one. ಈ capital T small t case ಅಲ್ಲಿ capital T is dominant over small t. Dominance is always tallness is always shown as dominance. So capital T is dominant over recessive. And in such a case, ಈ ತರ ಬಂದಾಗ ಏನಾಗುತ್ತೆ, ಈ ಸ್ಮಾಲ್ ಟಿ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗಲ್ಲ. ಮೋರ್ ಓವರ್ ಈ ಕ್ಯಾಪಿಟಲ್ ಟಿ ಎಕ್ಸ್ಪ್ರೆಸ್ ಮಾಡಕ್ ಬಿಡಲ್ಲ. ಓಕೆನಾ? ಸೊ ಕ್ಯಾಪಿಟಲ್ ಟಿ ವಿಲ್ ಎಕ್ಸ್ಪ್ರೆಸ್, ಸ್ಮಾಲ್ ಟಿ ವಿಲ್ ನಾಟ್ ಎಕ್ಸ್ಪ್ರೆಸ್. ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಲಾ ಆಫ್ ಡಾಮಿನೆನ್ಸ್. ಒಂದು ಡಾಮಿನೆಂಟ್ ಆಗಿದೆ, ಒಂದು ರೆಸೆಸಿವ್ ಆಗಿದೆ. ನಿಮ್ ಕ್ಲಾಸ್ ಇರ್ತಾರಲ್ಲ ಬ್ಯಾಕ್ ಬೆಂಚರ್ಸ್, ಫ್ರಂಟ್ ಬೆಂಚರ್ಸ್. ಬ್ಯಾಕ್ ಬೆಂಚರ್ಸ್ ತುಂಬಾ ಫುಲ್ ಮಾತಾಡ್ತಾ ಇರ್ತಾರೆ, ಫುಲ್ ಮಾತಾಡಿ ಫುಲ್ ನಾಯ್ಸು. ಕ್ಯಾಪಿಟಲ್ ಟಿ. ಫ್ರಂಟ್ ಬೆಂಚರ್ಸ್ ಸ್ವಲ್ಪ ಕ್ವೈಟ್, ಫ್ರಂಟ್ ಬೆಂಚರ್ಸ್ ಸ್ವಲ್ಪ ಕ್ವೈಟು, small t, quiet. Okay, so one is very noisy, one is very quiet. Now law of dominance is used to explain the expression of only one of the parental characters. Only one of the parental characters yavaga in the f1 nodi, f1 ilagittu. In f1 only one of the parental character is expressed. But tallness matra express agide, dwarfness is not expressed in your f1. It is not expressed in f1. Okay, only one of the parental character is in a monohybrid cross in the f1, and the expression of both in the f2. How the nodi, in f2 what do we have? This is called as f2. In f2 we have both tallness and dwarfness. Both the parental characters are expressed in your f2. In your f2, tallness, dwarfness. Students, you observe madirbodu. In the case of your monohybrid cross, there is always a single character is involved. Single character matra. For example, ide example nodi. Ee example bari we are talking about only about stem height. Yes or no? We are only talking about stem height here, nothing else. So monohybrid. Okay. An expression of both in f2. It also explains the portion of 3:1 obtained at the f2 generation. This is called as law of dominance. F1 ಅಲ್ಲಿ one is expressing, other one is not expressing. Law of dominance ಅರ್ಥ ಆಯ್ತು. Now what is law of segregation? What exactly is happening law of segregation? Now students, if they ask you in examination, this is what you're supposed to write. But I will explain in detail also one more time. Okay. The law is based on the fact that alleles do not show blending. This is the most important point. That is, alleles do not show blending. Yavaga F1 alli. See, here I am writing capital T small t. ಒಂದು ನಿಮಗೆ ಎಕ್ಸಾಂಪಲ್ ಕೊಡ್ತೀನಿ. ನೀವೆಲ್ಲರೂ ಒಂದು ಓರಿಯೋ ಮಿಲ್ಕ್ ಶೇಕ್ ಮಾಡ್ತಾ ಇರ ಅನ್ಕೊಳ್ಳಿ. ಓರಿಯೋ ಮಿಲ್ಕ್ ಶೇಕು. ಓಕೆನಾ? ಅದಕ್ಕೆ ಫಸ್ಟ್ ಏನು ಆ್ಯಡ್ ಮಾಡ್ತೀರಾ? ಹಾಲು ಆ್ಯಡ್ ಮಾಡ್ತೀರಾ. ಮಿಲ್ಕ್, ಶುಗರ್, ಓರಿಯೋ ಬಿಸ್ಕೆಟ್, ಐಸ್ ಕ್ರೀಮ್, ಮಿಕ್ಸಿ. ಅದಾದಮೇಲೆ ಆಚೆ ಮಿಲ್ಕ್ ಶೇಕ್ ಬಂತು. Now, is it possible to separate the biscuit out of it? Is it possible to separate milk, sugar and ice cream out of it separately? ಆಗತ್ತೆ ಯಾವುದಾದ್ರೂ ಮಾಡೋಕ್ಕಾಗ್ತಾರ? ಆಗಲ್ಲ. That is called as blending. ಅಲ್ಲಿ ಬ್ಲೆಂಡ್ ಆಗ್ತಿದೆ ಎಲ್ಲ. Okay, but in the case of your cross, in the case of your monohybrid cross, in the F1 generation, the alleles, the two alleles, capital T small t, just because tallness is expressed here, just because tallness is expressed in F1, it does not mean ಅದು blending ಆಗ್ತಾ ಇದೆ. It is not blending at all. The alleles are present separate. It is just that this one is not expressing, this one is expressing. So alleles do not show blending. ಎರಡು blend ಆಗ್ತಾ ಇಲ್ಲ ಇಲ್ಲಿ. ಸರಿನಾ? ಅಕಸ್ಮಾತ್ blend ಆಗ್ತಾ ಇದ್ರೆ, if it was blending, then this dwarfness would not be possible. ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. ನಾ ಹೇಳಿದ್ನಲ್ಲ, is it possible to remove the ice cream from the milkshake again? No. If this was blending right here, this would not be possible. This would not be possible. The dwarfness would not be possible if the alleles were blending. So the law of segregation says that the alleles do not show blending, and both characters are recovered as such in the F2 generation, though one of is not seen in the F1. That is, in F1 we do not see dwarfness, but again in the F2 we are seeing dwarfness again. It is coming back to life again, dwarfness. So alleles are not blending. Okay. Though the parents contain two alleles during gamete formation, the factors or alleles of pair segregate from each other such that gamete receives a single gamete. Artha ಮಾಡ್ಕೊಳ್ಳಿ. I'll explain a very simple one. This is also called as law of purity of gametes. ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. Your law of segregation is also called as law of purity. Now why is it called as law of purity? It is called as law of purity because ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. ಪ್ಯೂರ್ ಅಂದ್ರೆ ಏನು? ಪ್ಯೂರ್ ಅಂದ್ರೆ there is only one trait available. Hello Sangeeta, welcome to the class. See this. During gamete formation, e point illi, during gamete formation from the parents, the alleles are separating into gametes. That is, each gamete is pure for a trait. Illi very important line. Each gamete is pure for a trait. E gamete is pure for tallness. This gamete is pure for dwarfness. This gamete is pure for tallness. This gamete is pure for dwarfness. Each gamete is pure for a trait. That is called as law of segregation, also called as law of purity of gametes. Gametes are pure for a trait. Okay, for a trait. Though the parents contain two alleles during gamete formation, during gamete formation, okay, during gamete formation, the factors or alleles of pair segregates from each other such that a gamete receives only one of the two factors. Gamete levattu a single factor matra irutte. Of course, a homozygous parent produces all gametes that are similar, while heterozygous produces two kinds of gametes having one allele of each in equal proportion. Homozygous agidre single gamete, heterozygous agidre two gametes. That's all. Law of dominance and law of segregation. Law of segregation states that alleles do not show blending and alleles segregate during gamete formation. Alleles segregate into gametes. And each gamete is pure for a trait. One gamete, capital T agirbodu, pure for tallness. Small t agirbodu, pure for dwarfness. That is called as law of purity of gametes, and there is no exception to this law. This law right here is universal law. It is a universal law. Okay. Now in the dominance law of dominance has exceptions. Law of dominance has exception. There are two main exceptions. One is called as your incomplete dominance. Other one is called as codominance. Codominance. These are the two exceptions to this law. Two exceptions to law of dominance. Ade law of segregation ge no exception illa. Law of segregation ge no exception illa. Okay. Now that was your monohybrid cross done. Okay. Monohybrid cross aito. Now the next thing which I'll teach you is called as your dihybrid cross. Ready na ellaru? Ready na? Dihybrid cross. Ready students? Ready? Yes. Dihybrid cross coming up now. Now students artha madkoli ee concept tumba easy ide. But questions will be difficult. Same case in your in the board exam paper tagiri. Every single year in the PUC exam alli ee question baratte. What is law of segregation? What is law of dominance? Explain with the help of a monohybrid cross. Or what is dihybrid cross? They can ask you what is dihybrid cross? Explain with the help of an example. That is it. Dihybrid cross. Again, what is the first step in hybridization, students? The first step in hybridization is collection of parents. Now again we have parents here. Parents. The parents here is capital R, small y. That is round and yellow. Now what is round and yellow, students, here? Round and yellow are the two different characters here. In the monohybrid cross alli, there was a single character. But right now, in the case of dihybrid cross, we have two characters. That is seed shape and seed color. Two different characters here. Illi round nodi. The round is seed shape. Yellow is seed color. Both are dominant. Okay. Now we are crossing this with small r, small y. What is this? This is basically wrinkled green. Okay. This is basically wrinkled green. Okay. Now let's make gametes. Okay. Next step is gametes. Capital R, capital Y. Right? Capital R and capital Y. Okay. Now here also we'll get small r, small y. Okay. Small r and small y. Now here also this gamete is pure for one trait. This gamete is pure for one more trait. But only one trait will be there. Every gamete will have only one type of trait. Okay. Now what do? We find F1. Crossing F1. What do we get? Capital R, small r, capital Y, small y. Can anyone in the chat tell me what is the phenotype of this one? Anyone in the chat who can tell me what is the phenotype of capital R, small r, capital Y, small y? Anyone? A phenotype of this one. Very simple. See, capital R is dominant, capital Y is dominant. So this also will be round and yellow. This also will be what? Round and yellow. Round and yellow. Round shape, yellow seed. Okay. Next step is called as selfing of F1. Selfing of F1. Now students, in selfing of F1 we are taking capital R, capital Y, small y, crossing with capital R, capital Y, small y. Now when you want to cross this, First step is to make gametes. Now students, many students find difficulty in this step itself. How do we make the gametes here, sir? Students, there are 4 different gametes we'll get here. And there's a small trick here. The small trick is very simple. Multiply this with this and this with this. Then multiply this with this and this with this. Now first step, capital R, capital Y, first gamete. Capital R, small y, second gamete. Small r, capital Y, third gamete. Small r, small y, fourth gamete. So this with this and this with this, this with this and this with this. And that's how we obtain 4 different gametes from one parent. Similarly, from this parent also we'll get how many gametes? 4 gametes from here, 4 gametes from here. So in total we have how many gametes now? 4 into 4 and other side also 4. So 8 gametes are obtained here totally. Okay. Now students, let's do the cross. Let's do the cross. Now again here also we need to do the Punnett square. Again we need to do the Punnett square here also. Punnett square. Okay. Now here the Punnett square is going to be a little bigger Punnett square. Understand? First, what do we have? Capital R, capital Y. Capital R, capital Y. Capital R, small y. Capital R, small y. Small r, capital Y. Small r, capital Y. Small r, small y. Small r, small y. Okay. This is from one parent. This will also come from other parent also. So, capital R, capital Y. Capital R, small y. Small r, capital Y, and small r and small y. Now next step is to do cross. Simple cross. Let's start. Let's start, students. Let's start now. Okay. Now what do we get here? Capital R, capital R, capital Y, capital Y. Capital R, capital R. Capital Y, small y. Capital R, small r. Capital Y, capital Y. Capital R, small r. Capital Y, small y. Okay. Capital R, capital R. Capital Y, small y. Okay. Capital R, capital R. Small y, small y. Okay. Capital R, small r. Small y, capital Y. Capital Y, small y. Always capital first. Capital R, small r. Small y, small y. Okay. Capital R, small r. Capital Y, capital Y. Capital R, small r. Capital Y, small y. Small r, small r. Capital Y, capital Y. Small r, small r. Capital Y, small y. Okay. Capital R, small r. Capital Y, small y. Capital R, small r. Small y, small y. Small r, capital Y, small y. Small r, capital Y, small y, small y. This is your complete dihybrid cross on your board right now. Now after this you get, next step is to find phenotypic ratio and genotypic ratio. What is the phenotypic ratio first of all? Understand. Okay. Now you tell me, all of you tell me. What is the phenotypic ratio of this one? Round, yellow. Round because of R. Y, capital Y is because of yellow. So round yellow is the first type. Okay. Round yellow is the first type. First type which we get is round and yellow. Round and yellow. Let's see how many do we get round and yellow first. Okay. This is round and yellow. Let me draw some round yellow here. Okay. Round yellow. Okay, round yellow. This also will be round and yellow. Capital R, capital Y. Capital R, capital Y, round and yellow. Okay. Capital R, capital Y, round yellow. Now, this one you see. This is round, but small y, small y. So this will be round green. Yes or no? This will be round and green. Round and green. Okay. Next one. Capital R, capital Y, round yellow. This one also round green. again round green, round green. Capital I, capital Y, round yellow. Capital I, capital Y, round yellow. This one you see now. Next one. This one it says that, this one says that small r, capital Y. Small r means what? Wrinkled. Wrinkled yellow. It is basically now wrinkled yellow. Let's put triangle here for this. Wrinkled yellow. This also is wrinkled yellow. Small r, no? Wrinkled yellow. Round and yellow. Round yellow. This one you see now. Round and green. Round and green. Wrinkled yellow. Wrinkled yellow. Wrinkled yellow. Last one you see, small students. Small r, small r, small y, small y. What will be this one? Wrinkled green. So I'll put star here. Special category. Special category. Okay. Now students, tell me, how many yellow do you see now? Round yellow do we see? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 we see? Okay, we see 9. What is the next category? Round green. Okay, round green. Round yellow, round and green. Okay, how many do you see? 1, 2, 3. Okay, round green is 3. Now what is the next category do we see? Triangle. This is wrinkled yellow. Wrinkled yellow. Wrinkled yellow. How many do you find here? 1, 2, 3 triangle. Last category was wrinkled green. Only one. Wrinkled green. Only one. Here. Only one. That is wrinkled green. Only one. Okay. So we write 1. So do we get 9:3:1 ratio? Phenotypic ratio is going to be 9:3:3:1. That is phenotypic ratio. Now, in examination, this directly they can ask you how to derive this in board examination, or in KCET they can ask you what is the phenotypic ratio directly. They can ask you. Now, they will never ask you this. Generally, they will never ask you this in any of the examinations, the genotypic ratio. But I will teach you today. Extra information. No. What is the genotypic ratio? What is the genotypic ratio? Now there's a small trick here or hack I always teach students. I teach this. Now students, remember, your dihybrid cross is technically two monohybrid cross coming together. What do I mean by that? Tell me now. What was the phenotypic ratio of monohybrid? Phenotypic ratio of monohybrid was how much? Yeah. 3:1. Okay, 3:1. Now I told you dihybrid cross is basically 2 times monohybrid. So what do? Just try multiplying the students. 3:1 into 3:1. 3 3s are 9. 3 into 1, 3. 1 into 3, 3. 1 into 1, 1. 9 3:1, 9:3:1. Done. Now same technique can be used. Same technique can also be used to find the genotype of this thing also, dihybrid. How? Tell me, what was the genotypic ratio in the case of monohybrid? Genotypic ratio in the case of monohybrid. 1:2:1. Okay. 1:2:1. 1:2:1. Now multiply one more time. 1:2:1 multiplied. What do we get? 1 into 1 is 1. 1 into 2 is 2. 1 into 1 is 1. 2 into 1 is 2. 2 2s is 4. 2 into 1 is 2. 1 into 1 is 1. 1 into 2 is 2. And 1 into 1 is 1. This right here, student, this right here is the genotypic ratio. This right here is the genotypic ratio of your dihybrid cross. That's all. Small hack to find the genotypic ratio. I always teach my students. Okay, small hack. Okay, so it's given here also in your NCRT. Entire thing is given. This is the entire diagram. Phenotypic ratio 9:3:3:1. Phenotypic ratio. Okay. Now, students, with this dihybrid cross, the new law which you learn is called as law of independent assortment. Now, what is this law of independent assortment? Understand. Law of independent assortment says that in a hybrid, ನಿಮ್ದು F1 ಲಿತ್ತು ನೋಡಿ, F1 ಲಿತ್ತಲ್ಲ hybrid ಇಲ್ಲಿ. In a hybrid like this, when two characters are involved, segregation of one character, that is segregation of round, is independent of segregation of seed color. Seed round shape separate ಆಗೋಗುತ್ತೆ, seed color separate ಆಗೋಗುತ್ತೆ. The two characters will segregate separately. It is here. When two pair of traits ಇಲ್ಲಿ, one pair of trait is round and wrinkled, other pair of trait is green and yellow. Two pair of contrasting traits here, but right now here we have round and yellow. Okay, are combined in a hybrid, segregation of one pair of characters, that is segregation of round, is independent of segregation of yellow and green. Okay, seed shape will segregate separately, seed color will segregate separately. They do not blend. Okay, of one pair of characters independent of other pair of characters. Now, for the same reason we have four different types of gametes in your F2. Okay, now students, in examination they can ask you such questions also. See this question now. It might seem difficult, it's very easy. In a dihybrid cross, see, they're asking you dihybrid first of all. Okay, between a true breeding round yellow seed and true breeding wrinkled green seeded plant. Now, till here it is sounding like a dihybrid cross. Yes or no? Till here it is sounding like a dihybrid cross. Okay, but what is the actual question? The actual question is the ratio of segregation of round and wrinkled seed trait in F2 is. But in the end they're focusing on what? In the end they're focusing on only one character. See, starting they're telling you two characters and confusing you, but later on the question is asking you the ratio of segregation of round and wrinkled in F2, that is round, that is capital R, and small r, segregation of these two. Now students, a single character means what? Monohybrid. Now question is very simple. Now at the end the question is asking you segregation of monohybrid cross of these two characters. Okay, if there are two characters, one is dominant, one is recessive, this is a monohybrid, the phenotypic ratio is 3:1, and this is the answer. That's all. Okay, so such question can be asked in examination sometimes. See this question, all of you. In a garden pea plant, round shape of seed is dominant over wrinkled shape. All of you know that. A pea plant heterozygous for round shape. Okay, heterozygous, capital R, small r, heterozygous for round shape. Okay, is selfed and 1600 seeds are produced during the cross and subsequent germination. How many of the seedling would have the parental phenotype? Okay, now the question is asking which of the present will be parental phenotype will be present. Now here we have, this is the F1. Now if you do selfing of F1, what do we get? Capital R, small r, capital R, small r, capital R, capital R, capital R, small r, capital R, small r, and small r, small r. Okay, they're asking you how many of them seedling would have parental phenotype. What is the parental phenotype? The parental phenotype is round. Round is dominant. Round. 3 will be round. Okay, 3 out of 4 will be round into the genotype. What is the offsprings? 1600 offsprings. Okay, so do 3 by 4 into 600. You do how much? 3 by 4 into 1600. I will use calculator, but you can't use calculator. 3 divided by 4 into 1600 alla 1600. 1200 is the answer. C is the answer. C is the answer. Okay. Easy question. Okay. Look at this question now. Which of the following characters were not studied by the Mendel? Stem height, tall, short, seed shape, round, wrinkled, pod shape, inflated and constricted. Seed shape was not studied. So this is the answer. A is the answer. Which of the following characters selected by the Mendel in a pea plant is recessive character? Okay. Inflated was dominant. Green pod color is dominant. Axillary is dominant. But flower color, this is recessive. This is recessive. Why? Dominant is violet. Dominant is violet color dominant. So in examination, they can ask you such questions, students. In examination, they can ask such questions. It is very easy to solve, but also you need to think a little. Okay. And also let me know in the comment section if you want to solve more PYQs chapter-wise PYQs. If that is your goal, we will bring top 15 PYQs from this chapter next time when I come. Okay. Next question. In one of the hybridization experiment, a homozygous dominant parent and homozygous recessive parent are crossed. Homozygous dominant, homozygous recessive. It's a dihybrid cross. Okay, dihybrid cross. Dominant parent trait appears in both in F1 and F2? Yes. Generation recessive parent trait appears only in F2? Yes, this is the answer. Ille banta answer. Okay, read everything else, you will get to know. Okay. So, this was a very small session. This was a very small session to understand, give you a feel. ಯಾವ ತರ questions ಈ chapter ಇಂದ ಬರ್ಬೋದು. Okay, so let me know in the comment section which type of questions do you need the next time and I'll make it happen. Okay, so let's do PYQ, some questions in the next class. So with that being said, students, thank you so much. Comment ತಿಳಿಸಿ ಹೆಂಗಿತ್ತು ಕ್ಲಾಸ್ ಅಂತ, ವಿಡಿಯೋನ ಲೈಕ್ ಮಾಡಿ, ವಿಡಿಯೋನ ಶೇರ್ ಮಾಡಿ ಎಲ್ಲರಿಗೂ. Okay, so thank you so much, students, and I will see you in the next class. Bye bye. Take care all of you.